Good day everyone, I am Chinchina94. So for today's topic is all about year 2001 when cellular genomics and the Human Genome Project create a draft of human genome sequence. In 2001, the Human Genome Project International Consortium published a first draft and initial analysis of human genome sequence. The genome of any given individual is unique. Mapping the human genome involves sequencing a small number of individuals. Therefore, the finished human genome is mosaic, not representing any one individual. Human Genome Sequence was published by Nature and Science magazine. At the same time, Craig Venter and colleagues working at Cellular Genomics Corporation published another version of the human genome sequence. A wealth of information was obtained from the initial analysis of the human genome draft. For instance, the number of human genes was estimated to be about 30,000, later revised to about 20,000. Researchers also reported that the DNA sequence of any two human individuals are 99.9% identical. So guys, let's have some recap that in year 2001, Cellular Genomics and the Human Genome Project create a draft of human genome sequence. That's why I hope you learned something. Thank you for watching. Goodbye!
Good morning. Today I will discuss one of the events that happened in the history of biotechnology, which happened in the year 2002. So the event that happened in the year 2002 is rice becomes the first crop to have its genome decoded. So rice was the first sequenced crop genome, paving the way for the sequencing of additional and more complicated crop genomes. The rice genome was one of the few truly multinational plant genome projects, one with flags planted in chromosomes. One impact that the rice genome had, even in the midst of sequencing, was the first completely sequenced complex eukaryotic centromere on chromosome 8, which are usually gaps in genome sequences as they are highly repetitive regions of the genome. So, to put this all together, the availability of the rice genome, together with the community annotation and other resources that added functionally transformed genetics research and rice breeding. This can be measured any number of ways, but the increase in gene cloning is one direct measure. Ongoing work to understand the evolution and domestication of rice, to use this and information on genetic architecture, underlying plant physiological responses and phenotypes, is now being used to engineer superior rice strains to feed our growing population. That's it for my presentation about rice genomes. Thank you for listening.
Hi, I am Legislyn Macalindong, and for today we'll talk about the Human Genome Project. But first of all, what is a genome? A genome is the complete set of DNA including its genes. Now, the Human Genome Project provides information on the location and the sequence of human genes on all 46 chromosomes. It determines the base pairs that makes up the human DNA, which is about 3 billion. And they identify that there are about 20,000 to 25,000 genes in a human DNA. It is an international research coordinated by the U.S. Department of Energy and the National Institutes of Health. It began in 1990 and it was completed on 2003, which is 2 years ahead of its original schedule. So, is a genome again? Yes, it is the complete set of DNA, including all of its genes.
Hi everyone! I'm Maureen Kisha Mercado from St.Augustine and I will discuss about the Japanese experiment module, or also called KIBO. So, Kibo is a Japanese word which means hope or pag-asa. Kibo is a Japanese science module for International Space Station, ISS, and was developed by Japan Aerospace Exploration Agency or JAXA. So, contribution to ng bansang Japan sa ISS para mas mapalawak pa yung mga scientific research and studies. So, Kibo is a JAXA's first human space facility on the International Space Station, ISS, and is designed to host up to 4 astronauts to perform experimental activities here. So, isa tong pasilidad para dito i-perform ng mga astronaut yung mga scientific research, studies, mga knowledges, para kapag spread na nila yung knowledge, tamang-tama na. So, Kibo was also designed for scientific research activities on orbit and also enables educational, cultural, and commercial opportunities. So, madami talagang advantages ang Kibo na ito. So, Kibo was launched back in 2008. So, for our short assessment, the question is, what agency developed Kibo? Again, what agency developed Kibo? I'll give you 5 seconds to answer. Right, it's Japan Aerospace Exploration Agency or JAXA. Hope you learned a lot in this video. Thank you!
Hi, my name is Aaron Clyde Sambulia and I am from St.Augustine of Lipo. And now I am discussing the history of biotechnology 21st century in 2009. St.Augustine is widely known for its national leadership in transforming healthcare for the benefit patients. St.Augustine is a leader in the clinical care and research of heart disease, cancer, and brain disorders among other areas. In 2009, St.Augustine Heart Institute uses modified SAN heart genes to create the first viral pacemaker in guinea pigs, now known as ESANs. St.Augustine Heart Institute researchers have reprogrammed ordinary heart cells to become exact replicas of highly specialized pacemaker cells by injecting a single gene, or TBX18, a major step forward in the decade-long search for a biological therapy to correct erratic and failing heartbeats. And that is the history of biotechnology 21st century in 2009. Thank you!
Hi there! My name is Sam Lawrence Luico of St. Augustine of Lipo. And today, we're going to discuss one of the important events of biotechnology. Let's proceed, shall we? In 2012, a man named Zach Votter used a bionic leg that you can control using your mind to climb the Chicago Wills Tower that has a height of 442 meters. If you are wondering how does this connect to biotechnology, let me explain. If you remember, the leg he used is a bionic leg, and bionics is an engineering that applies biological methods and systems found in nature. Now, I'm going to ask you some questions. I hope you can answer them. Let's proceed, shall we? Question number 1. Who was the man who climbed a building using a bionic leg? Correct. The answer is Zach Votter. Next question. What building did Zach Votter climb? Correct. The answer is Chicago Wills Tower. Next. How tall was Chicago Wills Tower? Correct. The answer is 442 meters. And for my final question, what year did Zach Potter climb the Chicago Wills Tower using a bionic leg? Correct. He climbed the Chicago Wills Tower in 2012. I hope you learned something today, and thank you.